Hey Dan. Hey Dan.
Hi, Anton. Can you hear me?
Yeah, I can hear you.
Let me see that I put my webcam on. Does this work?
Yeah, I can see you. I have quite a lot of people in the waiting room.
Okay.
98 people.
Ooh.
Yeah.
Okay.
So once you're ready, I will hit admit all to start them. Okay.
Okay, that's good. Yeah.
So you will present from your local Jupyter notebooks, right?
I actually made a presentation for this.
Perfect.
I thought I was going to make a presentation, but yeah, I can do both. I can also scroll a bit through the Jupyter notebook.
Yeah. Maybe you switch a bit to code. Yep.
Yes.
Okay.
Yes.
So
Okay, sure.
Yep.
Let me also just put on the timer.
Mm-hmm. Right. So now we have 100 people allowed by Zoom. This is maximum. Actually, there were almost 2,000 people wanted to view this event. So yeah, it's true. So this is huge. So I'm recording this now and I will publish it to YouTube maybe so everyone can enjoy this session. Okay, so guys, let me introduce myself and then I have created this course together with them under the House of Europe organization grant. So it's sponsored by European Union. And we have created this course about data analysis in ecology on ecology data samples. And Dan was helping me. Actually, he almost completely created this course. I was just like an organizer of this process. So I will let them take over. And after this, I will show you the actually website where we have published all materials. I will put this link in chat so everyone can visit and see the codes and the lectures, the text on these topics. So Jan, please take over. Dan, sir, please take over.
Okay, wonderful. Wow, it's amazing to see that so many people wanted to join us and we can share with so many people the stuff we built. So yeah, let me just see if I can share my screen. Anton, can you allow the sharing? That would be great. Oh, you're also muted, Anton.
Oh, sorry. Yes. I will make you a host so you can share your screen.
Also wonderful.
Because I can't see this option. Yeah. Just try now, please.
Yes, just clicking around share. Wonderful. Okay, yeah, let me just start off the presentation then. Here we go. Hey, okay, so I think this should do the trick, right, Anton?
Yeah,
I think I'm in presenter mode. and
Yes, we can
everything?
see a screen. Right.
Hey, wonderful. All the nice technology these days, right? After 2 years of working from home, we're getting the hang of this. Okay, so everybody, super welcome today to the lecture. I'm seeing daylight, I'm seeing nighttime, so I think people from all over the world are joining, which is really nice. I'm guessing by the popularity of the course that a lot of people, well, it's super cool to see that so many people are interested in the course that we're going to provide, and that there is kind of a gap we need to fill still with the course we produced. So I'm hoping you will get to enjoy the presentation and get to enjoy the course. The files on the website Anton has built. So I'm really looking forward also to your feedback. You can always reach out to me to ask more questions or tell us that you're missing something important, and then we can see if we can still work on this. So before we actually dive into the community ecology part, maybe it's nice that I introduce myself first. So I'm Daan Mertens, and I'm a postdoc working in the laboratory of entomology in Wageningen in the Netherlands. That's also where I did my PhD work. And almost all of the stuff that I've been doing is on the interactions between plants and insect community. So mainly focusing on the herbivore part. That means that I'm definitely more of an ecologist than I am a data scientist. So you are much more likely to see me like this, hidden under a plant looking for bugs or covered in dirt, rather than behind a computer. And this is also kind of the perspective that I've been taking with developing the stuff that I've been writing. It's mainly focused on an ecological point of view, or teaching it to ecologists rather than to data scientists, because that's also where my teaching experience lies. I've been teaching a few ecologists, people who have started their PhDs. So I hope also you all experience it like this, that it's accessible for ecologists rather than data scientists per se. So in preparation of this lecture, I quickly Googled what community ecology actually is. And one of the first hits is, of course, a Wikipedia page, which gives you a very convoluted definition. So it's the study of interactions between species and communities on many spatial and temporal scales, including the distribution, structure, abundance, demography, interactions between coexisting populations. So that's a mouthful that actually doesn't say a lot, but let's break it down, and then we get to the essence why I think community ecology is actually quite a difficult science to be studying. So one of the first things that I want to highlight is that community ecology embraces the complexity of spatial and temporal scales. And this is something that is quite inherent to community ecology. If you imagine an early day naturalist running around the world and looking at all kinds of biodiversity, you quickly notice that on a spatial scale, communities are very different, and you want to understand why. Also on a temporal scale, it's quite easy to understand how communities change. Just think of seasonality. And a clear example of this are alpine meadows. And I really like these examples because they show such an incredible turnover in diversity on small spatial and temporal scales. If you ascend a mountain, you will see that the communities you encounter depends on the altitude you're at. But the same meadow is very different in summer than it is in the middle of winter. So the spatial and temporal scales of community ecology are very important and are by definition included in the field, let's say. The second part of Wikipedia's definition that I want to highlight is the interactions between species and coexisting populations. And I have to say that I don't fully agree with this part, just because I think it's too narrow. If you think about this, of course, community ecology is about the diversity of species in different communities. And you would, for example, look at different beetle communities in one side and look at the beetle communities in another side, and then you compare the diversity. That's a traditional view of biodiversity and community ecology. But it's not restricted to the species level. On many occasions, we actually don't have the information on the species. Take, for example, pollen samples. They are the beautiful spheroid picture on the left. They are very typical and characteristic for a plant species or a plant genus, but not always the full resolution, species resolution is available. Sometimes pollen can only be identified to the genus level. However, there is no reason why you could not do community analysis type of research on genus rather than on the species level. Similarly, the species concept might actually not be very appropriate to use. And then you could think of microbial communities. Microbiologists will not work with species, but with operational taxonomic units, the OTUs, so to say. There's no reason why you could not do very good community ecology without looking at the species. And similarly, if you're interested in diversity of genotypes rather than species, you could look at these butterflies, for example. They have very different morphology depending on the genotype. Well, there's no reason why you could not compare populations, the diversity of genotypes within populations using the tools that are developed in community ecology. And these were all very biological point of view perspective, but anything you can imagine that has a diversity component to it can be used in community ecology. And here I give the example of phytochemistry. So these are the chemical components that are produced by plants. Well, you can perfectly summarize and analyze chemical diversity of plants using community ecology type of approaches. So this is what I want to emphasize with, I think that the definition on Wikipedia is a little bit narrow. Actually, you can just stretch this to whatever concept you think is suitable to be analyzed by these tools. And the final part of the definition that needs some elaboration is this convoluted part with distribution, structure, abundance, demography, and then the interactions of species. And actually what we mean here is why do we find different species and different types and different numbers of species in different spaces or over different periods of time. And this is actually a core question in community ecology. This can be very obvious. If I would ask you to go look for a community of penguins, very few of you would go to the Sahara, most of you would go to the Antarctic, which is very obvious. But it's not the obvious cases, of course, that pique our interest. It are the less obvious patterns in diversity that are interesting to investigate. And these play at all sorts of different scales because ecology works at all these different scales. You can think of biogeography patterns, evolutionary history, environmental conditions, either biotic or abiotic, the population dynamics, chance effects, and so on and so on. And actually, fundamentally, what we want to know, whether you're looking from an applied perspective or a fundamental perspective, is which of these ecological processes are important in driving the similarity or differences in communities. Just to give an example, if you're a conservation biologist and you would want to know what ecological processes are causing your species that you're interested in conserving to go extinct, or why does this species occur in a specific area but not in another. These are the typical kind of processes you want to discover using community ecology. So overall, I think community ecology is a very elastic concept, but it's difficult to study simply because we're talking, each of us, about our own study systems in our own space and in our own time scale. So that makes that community ecology studies are often very difficult to compare. And that patterns that we discover using community ecology are also hard to generalize. An additional problem is that ecology is something that is studied within an environment, and typically processes that are on a larger or a smaller scale than the experiment that you're developing are important in shaping these patterns. So you have kind of a context dependency or a noise factor in your analysis. And this also means that patterns will be hard to generalize. And finally, but also very importantly, in my opinion, is that replication in all in all of ecology is quite problematic. And this is because you have this spatial and temporal dependency of patterns. In many cases, replicating studies is also very costly or unfeasible. For example, if you're interested in studying the community of species that live in a forest, yeah, it's very hard to replicate the forest yourself. It's extremely costly, also unfeasible. And for example, if you're studying the effects of pollution, I think you would all agree that it's kind of unethical to pollute a river just because you want to study the effect of pollution on communities. So you have a problem with replication. There is a reason why a river was polluted in the first place, right? So it might be that it has a nice flooding area where industry can be located. So there are all sorts of confounding factors that cause your study to be ill-replicated, or at least biased in its replicates. So again, this means that the patterns you find can be hard to generalize. So to sum up the introduction a little bit, what I wanted to point out is that community ecology, rather than being a very strict field of study that is well defined, I would think of community ecology as being a framework or a toolbox that has all different types of approaches and central ideas that can be used on whatever you would want to study, be it species or populations or chemical components in plants. You can apply it very easily. And it can be applied over spatial scales and over temporal scales. So it's a very elastic concept. But in general, these patterns are hard to generalize. There are no general laws in ecology, so to say. And because we are all working with our own specific scale and our own specific study organism, many tools have been developed. But that also means that the amount of tools that are out there can be quite overwhelming, and that there is the difficulty of choosing the right technique for what you actually want to show. So yeah, this is more or less what we try to address with the course, is give you a very solid base on which to build your own expertise as a community ecologist or ecologist in general. And we provide a lot of resources and extensions. So we discuss the basics and give you literature on how to proceed within your own field. And within this lecture, I want to focus a bit on the community ecology chapter. And that is focused around a seminal case study. It's a very textbook case study that's been used in different books already on multivariate and ecological analysis. It's a case study that has been conducted on a Wadden Sea island. And in the Wadden Sea islands, it's mostly coastal dunes with a bit of grassland on them. And you could sample the vegetation in different meadows on this Wadden Sea island. The meadows that were sampled differed in the way they are managed. So either they are used for standard farming practices, they are biological farming practices, some are used for hobby farming, and some are used for nature management. And the researchers just went to different meadows on the island and sampled a 2x2 square meter plot and recorded all the plant species that occurred in them. And the goal ultimately was to explore and describe the relation between the vegetation and the meadows in these meadows and the local management that was applied. So typically a community ecology data set looks somewhat like this. So you have a site by species matrix and a site by environment matrix. And the site by species matrix is simply all the different meadows these researchers went to. They are represented in the rows. And then in the columns, you can see the different plant species that were observed. The numbers within this matrix represent the abundance. So for example, in meadow 1, there was only one plant of Achillea millifolia recorded within that their 2 square meter quadrant, and so on. The additional information on the meadow, so which management type or any other information that you want to gather, in this case also the use of the meadow, whether it was used for hay production or as pasture, or the thickness of the top layer, or whatever environmental variable you want to record and use to explain differences in community, you can record in a site by environmental variable matrix. So this is typically what your community ecology study will look like. So let's go to the very, very basics of communities, and that is simply species richness and species abundance, right? So you can simply count the number of species within a sample and count the number of individuals you observe, and that's already quite a decent measure of your biodiversity that you can compare among the different sites. Very simple graph to make is just doing something like this, where in the x-axis you can see the different categories of land management. In the y-axis you can see either richness, that's the left plot, or the abundance of insects, plants, that's the right plot. And then you can see some patterns going on there. Yeah, so you could easily compare this already. But now we have to take one step back. What do we actually mean when we say that we want to compare richness across the different sites as a measure of biodiversity? Do we mean the total number of species that are possible? Or do we mean the total number of species that we can see in meadows of the same type? Or what do we actually mean with this? And this is a first extension of species richness that shorthandedly can be referred to as the alpha, beta, and gamma components of species richness. It sounds a bit complex, but it's actually very simple. Let's take these three meadows that are presented in the blue circles here, and they each have a set of plant species that occur them. So you could just count the number of species in each meadow. Yeah, so the first meadow has 4 species, the second meadow has 2 species, and the third meadow has 3 species. Yeah. They all belong to the same management type. So they were all used for example, hobby farming. Well, the total richness of plant species you could find in meadows that are used for hobby farming is what is called the gamma diversity. So this is the grouping variable. So you could think of all meadows that are managed in the same way. Well, these are the species you could find in them. That's gamma diversity. So back to the graph that I presented before, you could plot the gamma diversity on top of your alpha diversity. So how many plant species do you find in differently managed meadows? Well, for example, for hobby farming, that's the green one, the second one. You can see that the gamma diversity, so the total number of species you could find in these meadows is quite high. It's, I think, 22 from the top of my head. Well, it's higher than 20 at least. While for biological farming, this seemed quite low. It's actually 16. So these are represented by the triangles. If you look at how many plant species can you find in the different meadows that belong to the same management type, those are represented by the violins and the dots you see. So they are for let's say biological farming, maybe 9 species, 10 species, and 12 species. We have 3 replicates of that. So that's the difference between alpha and gamma diversity. Now, the next question we can ask is, how does this richness that you see within a single meadow compare to the total number of species you could find for all meadows of that type? And that's called beta diversity. So this is a sort of turnover example. So in other words, and I think this is the most easy definition of beta diversity, is which species are actually missing in your local meadow that could occur in your meadow. And in our case, let's say we had these species pool that could occur, the gamma diversity of 6 species. And we have a set of local managed meadows of 4, 2, and 3 species. Well, which species are missing? Well, in the first meadow, we are missing 2 species, so a beta diversity of 2. In the second meadow, we are missing 4 plant species, so a beta diversity of 4. And in the last one, we're missing 3 plant species. In the graph, you could actually easily summarize this as the difference between the gamma and the alpha diversity. So this is an additive definition of beta diversity. So the difference between the total species pool and the local species pool. However, this is not the only definition of beta that is out there. You could also use it as a proportion. You could also say, okay, this percentage of species or this proportion of species is actually missing from my total species pool. Which one definition you prefer to use of beta diversity really depends on your research questions. And also here already we are referring to literature because there is a huge diversity of beta diversity indices. And I would really recommend you looking into the paper by Anderson et al., which is a really nice overview of different types of beta diversity and why they are used. So yeah, this is a basic introduction to the alpha, beta, and gamma components of species richness. So one of the problems that we've actually kind of ignored until now is that we assume that for the meadows that we've sampled enough meadows. So we assume that all the species in each one of the meadows has been sampled and that none of the species are missing. And then this is a very valid comparison we could make, right? The problem is that if there are a lot of species, we also need a lot of samples to detect all of them. Because if I only sample 20 plants and there is a species diversity of 50, then I will never record all species that are there. This is also directly related to the abundance of individual species. If a species is very uncommon, then I need to take a lot of samples before I will be able to detect it. So I underestimate the species richness. Also the time spent surveying. If I would survey my meadow in autumn, or I would survey the meadow in spring, or I would sample in both, then I would find a different species richness for the different seasons. Also the size of the area. If I sample a 2 square meter or the full meadow, you can imagine that I would find a larger number of species if I would sample more intensely, if I sample a full meadow. So this assumption of us finding all species that are there is not very valid. And this leads to an estimation bias in species richness, which we will need to correct for. Because, yeah, if I make a mistake in calculating the species richness for biological farm meadows, and I make a different bias for hobby farm meadows, then comparing these two measures of species richness is not going to work. It's not going to give me a very valid result. So this is where species accumulation curves come in. They're actually quite straightforward, unlike richness, because I started by saying that richness is a very straightforward statistic, but apparently it's really not. It's very difficult to estimate species richness. And one of the key ways of correcting for a bias is by standardizing them. The samples we're taking. And this can be done with species accumulation curves. So species accumulation curves are easiest to understand when you think about sampling individuals. So let's say that I sample one plant in my meadow. Well, obviously, I'm going to find one species. If I now sample a second individual, well, there is a probability that this is going to be a different species than the first one I recorded, or the same species than what I recorded. Now I can repeat this exercise over and over and over and over again until I have sampled, let's say, 50 plants per meadow. Now, depending on how many species and how they are how abundant these species are, you will see that the curves, the number of new species I find with every plant that I observe, is going to start to slow down. If I sample all of the individuals in my data set, I can see that around 100 samples, I will have all species recorded for biological farming. It will take a lot more samples to find all the species in standard farming, for example. So this is the saturation point of the species accumulation curve. So based on these curves, we can ask some interesting questions, right? First of all, we can standardize the number of individuals that we sample and just say, okay, what would have happened if we compare the diversity of taking 50 plant samples in each of the meadows? So that's called interpolation. That would happen if we just take, for example, 50 individuals in each of the meadows, see how many species we find, and then we compare that. On the other hand, we could also do an extrapolation, and that's an extrapolation to the asymptote. And let's see at a certain point, how many species could we potentially have found if we sample infinite individuals. And finally, we could also standardize based on completeness. So if we would have sampled 40 or 50% of all species in the community, how many species would we have found? And then we can compare them across the different management types. So diversity indices are an extension to this. And then so maybe it's interesting to first have a quick look at how the Jupyter notebook looks like around now. So let me just click on this one. See if I can go there. Okay, so I hope you guys can see the Jupyter notebook now, or at least the text-based.
Yes, we can see.
Okay, wonderful. So this, until now, I've mainly been introducing the topic and introducing the dune meadow study. So this is all nicely written out in the course you can follow online and read through a bit more carefully. I also explore the case study, so showing you how the community data looks like and different management types that we're going to look like at. Introduce the topic of species richness and abundance, what we just discussed until now. And these are different graphs you can make. I invite you to have a closer look after the presentation, of course. Take your time to read through it and let us know what you think. And if you need extra information and feedback on this, would be great if we can get some info from you guys too. Then we switch to the diversity in alpha, beta, and gamma components, also what I just showed you before. And then we discuss diversity indices. So just like I told you before, that abundance is also a really interesting diversity metric to take into account. Like, it's not only how many species you have, but how many individuals of each of these species you have that you might want to take into account when describing a community. And this can be done by all sorts of diversity indices which correct for how abundant species are. Typically, or the most commonly known are the Shannon Diversity Index, the Simpson Diversity Index, and Berger-Parker Index, which is an index for dominance. So in view of time for the lecture, I will not go into too much detail here, but I really invite you to have a look at how they are calculated, how they are interpreted, and how you can calculate them. As you can see, the notebook also contains a lot of code, which is annotated. So you can just use the notebook to run R and run the data yourself, and then see what the graphs look like and whether you get everything, all the information in your R console. And then we go to the species accumulation curves that I just showed you. The importance of these species accumulation curves is that they give you a nice estimate or a more accurate, unbiased measure of species richness, which is the key statistic in many of these summarizing biodiversity indices. You want to be able to compare species richness across different treatments or management types before you can start calculating diversity measures and comparing them. And actually, there is a really cool package which is called iNext, which is in the code over here. And iNext is a sample-based or individual-based diversity measure. So it will give you, for every of the sites in our case, different management types, diversity metric, either species richness, Shannon diversity, Simpson diversity. It tells you what value you observed, what the estimation was, what the standard error around this was, and the confidence intervals, which are really nice to start comparing. And this is actually where you want to go to. And this is also what you can pick up from the course when going into more detail in the text, is that you can really start comparing these curves across the different management types and really start comparing communities based on their diversity. So because it's quite technical at a certain point, in the sense that it takes some time to read through. I will not go into too much detail, but it's a very powerful tool to compare diversity measures.
Then people asking to zoom in a little bit.
Sorry, of course. So is this better? Because for me it's zoomed, but I'm not sure whether it's for.
Yeah, looks better.
Yeah. Okay, but because I can still go a bit further. Okay. But I think, Anton, you will also provide the link to the website soon, right?
Yeah, sure. Website and all materials you can see after this lecture.
Perfect. Okay, so then I also invite all of you to have a closer look at the script yourself. And if you still have questions on any of this, feel free to post or email, and then we can either update the course if it's a very general question that seems to reoccur, or we answer specific questions. Okay, so let's see if I can switch back to the presentation now. Which should be here, I think. Oh no. Okay, so am I back in the presentation now, Anton?
No, we still see your notebook.
Okay, let me just try it a bit like this again. I'm not a host anymore, that's why.
Yeah, just give me a second. All right, here you go.
Yeah, perfect, thanks. Yes, I should be back, right?
Yes.
Okay. Okay, so this is exactly what I've been showing you also in the notebook really quickly, is that we've been looking at community data by summarizing them in univariate measures. So typically this is species richness, the abundance of species in your community, or any diversity index that you want to derive from them. And that's what you typically then would use in a regression type analysis. So this is the other chapter that I've created together with Anton, which I will not go into too much detail here, but these are what you would actually use to statistically analyze. And then you can compare communities with a statistical test. However, comparing communities is not so straightforward, because as you saw, the species richness was already very biased. It depends on how many samples you take, how intensively you sample, how complete the sample sizes were. And we provide you with a tool on how to adjust this bias using species accumulation curves. And your richness as a basic measure, it is not the only one that you could use. There are other diversity indices you could calculate, such as the Shannon and the Simpson index. And these really take into account how common species are, or how important rare species are for your studies. Okay, one of the biggest problems of analyzing communities in this way is that it's exactly what it is. It is a summarizing statistic. You are taking something that is very complex, a whole community with the abundance of many different species, and you are pressing all this information into a single metric, whether it's species richness, whether it's the Shannon index. In any case, you're using a lot of information. And this is why multivariate analysis is so important in community ecology. In multivariate analysis, the idea is that you try to contain as much information as possible with the species occurring in them. And then based on how similar meadows, in our case, are, you want to plot them close to each other, or identify that they are more similar in their communities. And a key step here is going from your community to a dissimilarity or a similarity matrix. And this is actually also not too difficult to understand when you break it down. So here we are back with the 4 meadows I already showed you before. These are 4 meadows that you can find on the Wadden Island, one with 4 species, one with 2 species, and one with 3 species. And rather than just saying how many species there are, we also want to say how similar these meadows are. Well, you could actually say, okay, between the first site, the left one, and the bottom one, well, 2 out of the 4 species that you can find are the same. So it's 50%, or 2 out of 4. The top left, or the left side, and the meadow on the right, well, it's only 1 out of 6. So only 16% similarity between them. And the bottom one and the top right, well, there's 25% similarity. So this is a very intuitive way to also represent communities, right? You could say how similar they are in their communities, and this is presence absence based, but you could adjust this for the abundance. You could say, okay, if it has a similar abundance of species, it's even more similar than just having the same species. And this is the central idea between similarity measures. Choosing what you decide as being similar or dissimilar is very important, because let's say that the Sahara doesn't have polar bears and Antarctica doesn't have polar bears because they live on the North Pole. Well, the Sahara and the Antarctic region would be more similar in terms of their abundance of polar bears. But it's not because polar bears are not present that the 2 sites are by definition more similar, right? You could argue that based on temperature, you would define the 2 polar regions as being more similar. So defining what you actually mean with sites being more similar in terms of their community is extremely important in community ecology. Unfortunately, it's also a little bit arbitrary because it completely depends on your choice and your research goals. So within the course, we elaborate on several of these similarity or dissimilarity, I should say, metrics, such as the Jacquard, the Sorensen, Euclidean distance, and the Hellinger distance, which are each of them specific ways of calculating the similarity among sites in terms of the community that occurs on them. So I again invite you to read through the course material in a bit more detail, because it's, you need to absorb a bit of information before you get a full grasp of this. But these are very commonly used diversity or dissimilarity measures. Now, the way you calculate similarity or dissimilarity, like I just showed you for the 3 meadows, well, you could actually represent this quite easily in a graphic display. Very familiar, or at least to most of us, should be phylogenetic trees. In essence, what you're doing in a phylogenetic tree is you take the genetic composition of one species or an individual, and you compare it to the genetic composition of another species or individual. And the more similar these species are in terms of their genetic composition, the closer they are plotted together on a phylogeny. Very dissimilar species are plotted far away from each other on a phylogeny. Now replace the idea of genes with species in a community, and replace the idea of samples, phylogenetic samples, with your sites, and you can easily create what is called a cluster analysis. So in here, you calculate how similar communities are in terms of their species or genes, if you want. And then communities that are very similar are plotted close to each other, while communities that are very dissimilar are going to be plotted very distant from each other. And that's exactly what you do with a cluster analysis. The thing is that there are different rules of aggregating clusters. So you could say that as soon as something is similar, one element is similar enough, you combine them or you pool them. Sometimes you want a more stringent way. And this has a big impact on how your cluster analysis will look like. So kind of unfortunate that it's arbitrary and completely up to you which type of analysis you use, but your use of what you define as being similar or dissimilar and the technique you use to cluster sites together is going to determine what your results look like. So our advice is also based on literature, look at the research question you want to answer, look for what is the effect of choosing different dissimilarity measures and clustering techniques, and then start comparing. Maybe it's time to again quickly show the notebook on this, because there we go into a little bit more detail. I think it's this one, right? Yes. So I think you should see the notebook again. So let me scroll down. So here's the similarity in communities concept that I just explained to you, how you could calculate how meadows are more or less similar in terms of their community composition, how they are can be used to calculate the similarity matrix, the different types of the similarity you can calculate. We go into a bit of detail there on what each of them characterizes and how they are used. And then we go to the cluster analysis that I just showed you. You can compare all these different techniques. So these are different techniques: single linkage, complete linkage, average linkage, Warts clustering, that have a different algorithm behind it. And you can easily code them and plot them and compare them. So here in the top left cluster analysis, you see the single linkage clustering pattern. While this is very, very loose. It easily aggregates sites in a cluster, while complete linkage, the top right one, very conservative. And you can already see that even with the same dissimilarity or definition of dissimilarity, but a different algorithm used for clustering, I get a very different cluster analysis. By the way, the numbers on the tip of these branches refer to the number, the row number in my community data. So each of these numbers represents a specific meadow. Okay, so this is cluster analysis. My screen is paused. Let's see. Okay, back to the presentation. So the next step that is very commonly, or next technique that is very commonly used in community ecology are ordination techniques. So they are actually fundamentally very closely related to cluster analysis. It's also a visualization of similarity or dissimilarity among meadows in terms of their community. But rather than using a grouping, so a clustering, they use a more continuous type of representation. So let's make this very, very plastic for you. Imagine that I have one species, Lolium perennis, and I want to compare the different meadows in our case study in terms of the abundance of Lolium perennis. Well, a lot of the sites actually don't have any of the species. Some of the sites have 2 individuals, some of them have 4, some of them have 6, and this is plotted on the x-axis. For now, you can ignore the y-axis. I simply included it because otherwise the dots would be plotted on top of each other and it would be hard to interpret, right? So the y-axis doesn't really represent anything right now. It's just the x-axis and the spread along the x-axis that I want you to focus on. So each of the dots in the graph represents a meadow. All the meadows with 0 abundance of Lolium perennis are plotted all the way on the left of my graph because they have 0 abundance. And all the plots with a lot of Lolium perennis in them are plotted all the way on the right. So plots that are very similar to each other are plotted close to each other. Plots that are very dissimilar to each other in terms of Lolium perennis are plotted far away from each other. Okay, so this was for one plant species. We can easily extend this to include 2 plant species. So here I included Trifolium repens. And now the y-axis does represent something. So there are a few sites where Lolium perennis and Trifolium repens are not present at all. So they are in the bottom left corner. There are a few sites where there is some Trifolium repens, but no Lolium perennis. They are on the top left. And then there are sites which have both species present in high abundance, and they are on the top right. So again, the dots represent the different meadows. And I colored the different dots based on their management type. So in this way, you can actually represent the variation in communities or the similarity in communities with simple X and Y graphs. Now, the core problem here is that I have more than 20 plant species. So I can make all pairwise plots of all different plant species, and I would end up with 20 faculty graphs. So that's insane numbers of graphs that I need to compare before I can actually take any meaningful interpretation out of the data. So that's definitely not an option to do, right? Now, this is where ordinations come in. So you have to realize that community data is highly redundant. That means that plant species tend to occur, or many plant species tend to occur in similar environments. So let's say that you have a very wet environment. Well, it is characteristic of a community, of a set of species, and these same species will be absent when they are in dry environment. So they give us the same information. And this is what ordination actually uses. It tries to find correlated presences or abundances of species. And let me make it a little bit visual over here. So in plot A, you can see the original graph we had with the 2 plant species, Lolium perennis and Trifolium repens. Now we could easily switch our axis and represent exactly the same information in plot B. We just shifted the axis a bit. Now, if we now rotate the axis to represent the most variation in the first axis, the x-axis, and a bit less variation in the y-axis, and then rotate it back to the normal Cartesian axis system that you know, in plot D, then we have what is called centered and rotated the PCA data. So these new axes that we draw that are representing the most variation in our data set are called PCs in this sense, principal components. So that's the aim of ordination. Try to minimize the number of dimensions that you need to present the most dissimilarity possible. You can see that on the in figure D, that most of the variation is represented by the x-axis, while only little variation is represented by the y-axis. Now, if you would ignore the y-axis, then we would not lose too much information, right? Now, these ordination plots can look quite overwhelming. And this is actually what a PCA analysis would look like on the dune meadow data. And I will walk you through it a little bit. So you can see that there are, again, differently colored points, and the points represent the different meadows in the case study, and the different colors represent the different management types. And points that are close together are very similar in their community, while points that are far away from each other are plotted are more dissimilar in their community composition. You can also see that there are a lot of arrows that we drew on the PCA graph. Well, they represent the direction of abundance of species. That means that meadows that are plotted very close to the tip of an arrow are going to have a lot of that species, while plots that are far away from the tip of the arrow typically don't have a lot of that species in them. Finally, you can see that the first two PC axes, so PC1 and PC2, are greatly helping us simplify the community data, because with only two axes, we can represent 30.57% of the variation plus 19.98% of the variation, which is much more than we could do with just the plotting the species next to each other. So that's the goal of simple ordination techniques. It's very important to be aware that this ordination type of graphs are exactly what they are. They are graphs. They are not a statistical analysis. So just the same way that a box plot is not a statistical test, it gives you an indication of differences between different treatments, but it's not giving you a test. It's not testing any hypothesis. It's not giving you a p-value, so to say. So that's the difference between multivariate statistics and ordinations, so to say. So before I move on to give you a little bit of permutations, let's again go to the Jupyter notebook, because for now I've really been focusing on what a simple ordination would look like. And actually, this is what a PCA, principal component analysis, would look like. But there are many different types of ordinations, such as correspondence analysis or non-metric multidimensional scaling. And these are all part of what is called unsupervised ordination. So we're not using any environmental information to explain variation in the communities. Just like with the PowerPoint presentation slides I just showed you, we start by introducing the idea of using dissimilarity or similarity in plots to make these ordination graphs. See, it's the same figure with Trifolium repens and Lolium perennis, introducing you how plots that are very similar in their community are plotted close together. We also discussed this idea of principal components analysis, where you first rotate, center, and then rotate the data, your axis, and try to extract all the information out of it as much as you can. And we go to the analysis steps you can take. So this is very easy to just run and read through, and this will give you a lot of information. This is typical for our output. It's quite uninformative if you don't know what you're looking for. But that's also something we take a lot of time for. We go through all the different components of the summary output, discuss what they actually mean, how you can interpret them in function of the data we present as a case study, but also by extension, your own case study. So if you would run a PCA on your own data, you would get similar outputs, and you can use the our step-by-step summary of the output on how to interpret your own PCA analysis. And then we make a plot. We create the plot by hand. We extract all the information you need from the PCA analysis, and we combine them with annotated code to create this figure by hand. You can, for example, if you're not interested in these PC scores and you actually are only interested in looking at the similarity in meadows, independent of what species are actually in there, you could just remove the arrows from your plot, which can be done here in the geom segment kind of statement that we make in the code. So just mess around with this and see if you can follow the code. And if anything is not clear, or if we seem to be getting a lot of the same questions on this, we will update and further annotate for clarity. So it's quite flexible coding in that sense. We also provide you with the idea of scree plots, which actually tells you how many of these PC axes you should interpret. Because in our example, we first had the 30.57% explained, and then 19.98% of the variation in communities explained, which is quite a lot. It's 50.55%. But that also means that nearly 50% of all the variation in communities among the meadows is actually not represented by our PC. So how many of these PCA plots or how many of these PC axes should you actually interpret before you can make a reasonable statement out of this? And this is what scree plots do. They give you kind of a guideline on how to do this. I suggest you read through this, but in the end, it shows that the first 6 PC axes that we create by rotating multidimensional data should be interpreted according to the scree plot cutoff value that we made. Okay, similarly, correspondence analysis, you will find out if you're not familiar yet that correspondence analysis is very, very, very similar to what principal component analysis does. So I will not go into too much detail here, but you can just read through the text after the presentation if you're interested in performing correspondence analysis. We also discuss the same type of output. We create the same types of plots, which you can adjust yourself. It's hard-coded, so it's not based on an internal wrapper function or anything. We just extract the data from the results and use them to plot the results. Another technique that you might be interested in, which we don't have time for to discuss, is non-metric multidimensional scaling. I think it's a very cool technique, so read through it if you're interested in presenting community data. It actually tries to represent as much variation as it can in 2 or 3 dimensions. Okay, great. Then we need to talk about testing for significance. And this will be the final part of my presentation. So let's see if I can switch again to the PowerPoint. Okay, permutations. So here is the first time, or this is the first time within the whole course that we're actually really talking about getting statistical tests done. This is the first time in the whole community ecology course that you will end up with getting a p-value. So I'm hoping most of you are kind of familiar with the idea of doing an ANOVA analysis, for example. In essence, you're just trying to measure the amount of variation that you see within a group and compare it to the amount of variation that you see between groups. What you hope to see is that the variation between groups is larger and significantly larger than the variation you see within a group. And this is called the sum of squares. This is typically what you would use as use sum of squares for. It tries to measure the variation within a group, compare it to the variation between groups. And based on this, you calculate an F ratio. And this F ratio can be compared to an F distribution and gives you the actual p-value. So in essence, it's just checking where most variation is. Now in multivariate analysis, because we're dealing with a lot of different response variables rather than just one response variable, this is exactly the same as what we're doing. We're trying to compare the amount of variation you see within a management type for us to the amount of variation you see between management types. Are communities in different management types more dissimilar than meadows that belong to the same management type? That's more or less what you're doing. Now, permutation analysis has an interesting way of approaching this, because if that's true, if management types are what structures the variation, then shuffling meadows to belong to different management types should mess up this proportion. You would find as much variation between sites that are randomly assigned. So here is an example of a simple permutation. What we did is just take the abundances or the community that we observe in a site. For example, the first site has now become the third site. And instead of the second site being biological farming, well, that community is now assigned to the management type of standard farming. You calculate the amount of variation you see within a specific management type, and you compare it to the amount of variation you see between management types. You calculate the F ratio of a randomized community matrix, and voila, you get a F value that you can use to compare. You do this tons and tons of times. Depending on the size of your study, this is 100 times, 1,000 times, 99,000 times. You see all sorts of random numbers, but you just keep generating shuffled communities. And then based on this, you get you each time again calculate an F value. And the p-value that is associated with this is the number of times you find that the F ratio, so the variation between groups, is actually smaller than the variation within groups. So for our case, if we randomly permute our community matrix a high number of times, you get a distribution of F values that is given by the histogram, while the one that is measured by us, the one we think management is going to be important, gives an F value that is close to 3, which is kind of on the outside of the possible F values of random matrices. So to summarize this a little bit, we calculated an F value based on our observations, which was here represented by the vertical line, close to 3. Then we randomized the community, and for random communities, we found a whole range of F values ranging between 0 to, reasonably around 2. So a random matrix has an F value of between 0 and 2, while our matrix, our community matrix, had a much higher F value, showing that management is indeed an important factor in structuring the communities belonging to different species. So this is what a permutation test or a MANOVA actually does. So from this, let's, before I go to the take-home message, quickly show you what the notebook looks like again for this section, because it's much more elaborate than what I've been able to show you within the presentation. So we discussed the MANOVA and PERMANOVA analysis, so the one that I showed you or discussed with you that is really about this variation within groups and between groups. In our case, the variation within meadows of the same management type, comparing it to variation between management types. And because we are dealing with non-normal data, we need to permute it. This is also what I showed you in the presentation. That's shuffling meadows to belong to different management types, calculating an F statistics, and comparing the distribution of F statistics that you calculate for random sites to the one you actually calculated for your original data. This is also presented in a figure, which you can tweak yourself if you are in need of getting permutation tests a bit more visual. You can do it by using this code. It creates for you an F statistic that you see, the horizontal vertical line over here, and a distribution, a null distribution, it's called, of possible values for random matrices. Okay. And then finally, there's also a topic on mantle tests, which works very similarly. But I will not discuss this right now. I advise you to look through the course for this and get some more information. We conclude the course chapter by talking about supervised ordination. And actually, they are kind of similar to unsupervised ordination. So the PCA and CA analysis that I've shown you before, except that they take environmental information. So land management types or soil moisture, whatever you have measured as environmental variable, and uses it to constrain variation. Exactly how this is done, you can read in the text here. There's also information on how to run them and how to interpret them. We again run through all the output of the summary function. So this should be clear once you read through it. We again make different plots and that are tweakable. You can adjust them to your own needs. And that's more or less it. This is more or less what we address in the community ecology course that is on the notebook. So to wrap up, I want to give you some take-home messages from the presentation I just gave. So one of the first things that we talked about is that community ecology is kind of a difficult science, not so much a well-defined field of science, as more a toolbox of different techniques that you can use. It's very elastic. You can use it on a study system, on all types of spatial scales, whether you're looking at the Rocky Mountains community or the flowers in your backyard, or whether you're looking at the bacteria in a petri dish. You can use these same tools. You can also use them over different time scales. You can use them over different types of biological organization, or even outside of biological organization. I gave you the example of chemical compositions. You could easily use community ecology to study or analyze diversity of chemical compounds, for example. So it's a very flexible and very elastic concept. One of the main problems with simple community ecology is that you are summarizing a lot of information. We gave the example of species richness, which by itself was already very problematic to accurately measure. But if you do it correctly, it's a very powerful tool because it's very easy to communicate. This site is more or less species rich than another site. You can easily use these estimations in a regression type analysis, do some statistics on them, and you get a really interesting, hopefully meaningful result for your study. So then moving on from univariate statistics or univariate community measures, I also showed you how communities can be summarized in terms of similarity. So the similarity or similarity by showing you cluster analysis and ordination techniques. These are far more elaborately discussed in the notebook. than in the presentation. Overall, I think with multivariate analysis, the trick is that it tends to be a little bit arbitrary which the similarity matrix you use, which type of visualizations you tend to use. Also, it's often more difficult to explain to people what you're actually doing in multivariate analysis. So it takes more effort than using a summarizing statistic like species richness or Shannon diversity. But I really advise you, if you're looking into using community ecology, to read through the course and really try and embrace multivariate analysis as a more comprehensive way of showing your data. And with this, I want to thank Anton for inviting me to collaborate on this project. It was really also a very steep learning curve for me on how to prepare this lecture and how to present the course material. But I personally think that the result is quite nice. So I'm really looking forward to your feedback on this. And also during this period, I've been supported by the lab in Entomology in Wageningen, mainly by Eric Poelman and his ERC grant. So that has been part of the funding. And also House of Europe, who is providing funding for this. Okay, and thank you.
Thank you very much, Dan. I see we have a couple of questions in chat. Maybe you read it. And,
yeah, so
on this. Yep, last few questions I see. Something about the H class method.
okay, let me just open the chat real quick.
Yeah.
Okay. Okay, so let me scroll through this. So the first one is on, I can see that there is a question for bibliography, and this one specifically on species accumulation curves. But actually, in general, I've tried to, throughout the notes in Jupyter, also refer to literature. You can just look in there, and then there are several paper references or book references that are, in my opinion, very good, or that I've used intensively for building the course myself. Now, specifically for species accumulation curves, I think I refer to, let me just go to the bibliography real quick. So there's a lot of work by the team of Gao et al. I will quickly post them in the chat down here. So these are two references that are really, really good. That's also the team that developed a standardized way of going for extrapolation. So extrapolating to species richness is actually also quite difficult. You have to have a certain confidence level around this. They really explain this very well. And they are also the people who built the diversity package. So the iNext package that you probably want to know. It comes from the same lab. It's absolutely great. Also well supported, so they have great resources available. So yeah, I suggest you look into these references for more specific information. Okay.
There is a question about feedback, how to apply feedback. Is it a pull request to Git or maybe another way? I think email would be the best way, because we cannot open Git repository to allow anyone to post fixes. Or maybe, I don't know, maybe it just will take too much time of Dan to review all pull requests from people. I don't know which is the best way, but let's prefer the email, I think.
Yeah, I would say prefer the email. You can also try to direct contact me on Twitter. I will post my my information. I hope I don't get crazy spammed on on my media. So I will take some time to answer them. I will post it here in the chat real quick. So
I have actually your Twitter on the website. I will post it
okay,
later.
wonderful.
So
Yeah,
don't worry. I have a page with authors, and there is your image and Twitter link.
perfect. Okay, so then you can reach out to me through any of the approved methods for sure.
Okay, let's move on. Facebook group is Ecology in R. Yeah, I see someone has answered. So the next question is, how can you know which method to use for cluster?
Yeah, so I think this refers to which clustering algorithm you should use. So that's kind of tricky because it's arbitrary. Any kind of clustering technique is valid. As soon as you have community data, you can use single linkage clustering or median linkage clustering or whatever you prefer. My advice to you would be with almost all visualization techniques is try and see what makes sense. Focus on your research questions. What do you want to see? Do you want to be very loose in your conclusions? As soon as anything is similar enough, link it. Or do you want to be a little bit more stringent? Does it have to be exactly the same? They also, within the course, I think I have a small paragraph on this, on which linkage technique to use. So my advice would really be explore them, plot them, see what is meaningful for your type of data, what gives results that are a bit matching with what you would expect from your species richness to look like, for example, or combine it with an ordination, or combine it with a heat map that gives you different options. And all of these should be linked within the notebook. You can just look up how to do these things in the text. And yeah, so just compare them, see what is interpretable for your research question, and go with that one. Just be very clear on what you decide to do. Just be very clear on which dissimilarity metric you use, which clustering algorithm you use. And if you're using it as in a paper or in a thesis chapter or whatever you want to use it for, always give a clear methodology. Be very descriptive in your methodology, because yeah, multivariate statistics, if you're not specific in which techniques you use, it's kind of a black box, right? So indeed, the function we use is hclust or hcclust, I have to say. But there are different packages out there. Like anything of these type of analyses in R, there are more packages than people. In the end, they're all tweaked to specific needs of the field. I think hclust is the most commonly used, and it's very user friendly too. So I would advise on that one.
Yep. Next question is quite long, and it compares this method to natural language processing, which I'm not an expert in.
So it looks like this is moving.
I'm not sure what's it about.
No, so that's definitely outside of the scope of the course. So it's very much no, I'm really going to skip this question because I actually don't really know enough about this topic to give a reasonable answer. So in essence, do you know of any work? I'm sorry, I don't know any work that would really use this. Sorry.
Right, and the last question is about cutoff line for cluster analysis. I think you have just answered it, so you should just explore it and compare all methods and compare the results. Right, I think that's it for today. Thank you everyone who has joined this session, and it's a bit pity that only 100 places in this room. So actually in this event, as I said, it was about 2,000 of people, but anyway, I have recorded everything and I will post all links and website and all the presentation files in this Facebook group. So don't worry if you have missed something, you will find it and can read it later. Right, and also thank you very much, Dan, for this huge presentation. It's really a wonderful work. I even didn't expect for such results. So yeah, thank you very much. You are great analyst.
You're welcome. You're welcome.
Right. All right. Thank you, everyone, and have a good day or good night. So goodbye.
Bye.